Hello students, welcome to my YouTube channel aksharaaalaya. As we continue to our final segment of microorganisms friend and foe, we will discuss about the three main topics of this chapter. That is how food preservation is done and why food preservation as a topic is included in this chapter of microorganisms friend and foe. You may think food is a different thing and microorganisms are different thing. Why food preservation is important and why it is included in this chapter. Next we will discuss about nitrogen fixation. As we have discussed in our previous segments, we will discuss nitrogen fixation, how it is done and which organisms, microorganisms are responsible for nitrogen fixation. Then to fix this nitrogen, as it is very inert and unreactive, to fix that nitrogen we need some microorganisms, and to fix the gaseous nitrogen to soil there is a cycle, that is nitrogen cycle, and nitrogen should also be maintained in atmosphere. How it is maintained in atmosphere? All the nitrogen is in the atmosphere, and if it is fixed to soil, how the percentage of nitrogen is maintained in the atmosphere, we will know that. First, going to food preservation, you may have seen your grandmother putting salt to the wrong mango pieces or wrong mangoes to pickle them, right? Is it only because of it gives a salty taste? No. You may have also seen your mother boiling the milk to a certain level that it raises its level and she will put it off. Then it goes to its normal level. What is the science behind it? Why she heats the milk in such a way just to enhance its taste and flavor? No. Both the techniques, knowingly or unknowingly, they are the techniques of food preservation. How they will affect microorganisms, we will learn that. As we know, food will be spoiled either by chemical reaction, that is by oxidative or reductive reactions, right? That is only some part of food spoilage. But foods also spoil because of presence of microorganisms. As we think food as a source of nutrition, microorganisms also for their survival think food as a source of nutrition. So they will invade the food material, they will extract the nutrients from that, and they will lead to food spoilage. To avoid this food spoilage, what we should do? We should avoid the growth of microorganisms. That is done by two main methods. One is chemical method and another one is physical method. As the name suggests, chemical means use of certain chemical compounds to preserve the food or some physical actions to preserve the food. Chemical preservation is done by adding salts. As I have said, your grandmother uses salt to maintain the raw pickles or raw mango for making pickles, right? Because salts will remove all the moisture content from the raw pickles or raw mangoes. That results in less moisture content. So in that condition, microorganism cannot live. There will be high salinity also, because salt is there, no salinity will be there. So microorganisms cannot divide and cannot survive in such conditions. So adding salt acts as food preservative. Then second one is adding of sugars. You may have seen the honey. Have you seen a honey getting spoiled? No. You may have also heard there are honey samples from Egyptian pyramids which are dating back to thousands of years back, but they are still fresh and ready to use till today, right? That is because they have high fructose or high sugar content and they have very less moisture content. In that case, sugars are used to raise the the sugar level and reduce the moisture level. Where the moisture is less, the microbe cannot grow in such condition. So sugars are used to prepare jams, jellies and all. Another is use of oil and vinegar, right? Your mother or anyone in your household have been using vinegar to preserve pickles and vegetables. Vinegar has been used, and mustard oil and such oils are used to preserve foods. Mustard oil is said to be very good preservative, and they have also been using it for many recipes in Indian cuisine. These are chemical. They use some or other chemical compounds like salt, sugar, oil, vinegar. Physical food preservatives are using heat or cold. We use cold temperatures, like we use refrigerators, right? Refrigerators that is below 8 degree to 4 degree Celsius, around that. And we use deep freezers below 4 degrees Celsius where the ice is formed. In such cold condition, microorganisms cannot live. There is no optimum temperature to live in such condition. So microorganisms division will be stopped, food spoilage will be stopped. And hot conditions, that is hot temperatures, boiling is also used. Boiling is used because when the temperature rises, the microorganisms start to die. In similar technique, there is another term called pasteurization. Pasteurization. As the name suggests, it was technique found by Louis Pasteur. Louis Pasteur found a technique to preserve the milk for longer time. In this technique, milk is heated for 70 degrees Celsius just for 15 seconds and then it is immediately cooled down. In that case, what will happen? Microbes present in milk will face a heat shock and they will die. When it is suddenly cooled, all the microbes will get dead. In such case, milk can be used for longer time. You may have seen the typing on the covers of milk, right? Pasteurized milk. Such milk is ready to use. You can directly use, no need to boil. But our mother boils it for extra security. What our mother does, how pasteurization is done in our house, our mother will keep milk till it raises its level while heating and she puts it down. It will reach around 62 degrees Celsius during that process and she will keep it around 15 minutes or 20 minutes, right? This is pasteurization done in lower temperatures with higher time. Not only cold and hot treatments, we use other treatments for preservation of food. That is, during storage and packing, we use sealed packs or where there is no gap left. Gapless seals are used for storing and packaging the food materials. As microorganisms enter the food material, we will avoid all the spaces which they can enter. So they cannot attack the food material. Once the food is boiled or added with salt, we will pack it, seal it to avoid interaction with microorganisms. When the food is freshly stored and packed, there will be no spoilage. So these are the some food preservation methods discussed in your chapter. So going forward, to learn about nitrogen fixation, as we know, nitrogen fixation is very important because nitrogen is a gaseous element, right? Nitrogen is a gaseous element and it is unreactive. It will not easily form any compounds and it will not be accessible to humans or plants. To make it an accessible compound, there is a need of fixation. It is done by a microorganism called rhizobium. There are many microbes that will fix nitrogen. One of them is rhizobium. This is associated with the roots of leguminous plants. Leguminous plants are those which have beans, peas as their outcome. Leguminous plants. The roots of leguminous plants will be associated with rhizobium. They both have a relationship helping one another. That relationship is called symbiotic relationship. Symbiotic means helping one another. For example, if you help your friend by giving a pencil or pen during his assignments or completing his classwork, he will return that help with a food, a piece of roti or a dosa during the lunch time. Such understanding or giving and taking mechanism follows in this relationship, that is between the roots of leguminous plants and rhizobium. What rhizobium will do? Rhizobium will fix the atmospheric gaseous nitrogen to the roots of leguminous plants. You can see the nodule-like structure. I have took a touch me not plant. I will show the nodules formed in touch me not plant. Okay, I will take it closer for you so that you can see it easily. So see students, you can see two nodules here. One is this, which is on my fingertip. Another one is this. This is a nodules of touch me not plant. Similarly, we will see the nodules in other plants uh, like peas and all. There are many nodules which are also covered in soil also. So you can see this here also. There are nodules which are covered by soil. Those nodules will have rhizobium bacteria or other nitrogen fixing bacteria. You can crush those nodules and see under a microscope to visualize how the microorganisms look. So this rhizobium will fix the nitrogen. In return, leguminous plants will give them shelter and other nutrients required for the survival of bacteria. That is how they will help one another. But if they fix all the atmospheric nitrogen to the soil, is it possible to maintain the level of nitrogen in atmosphere? No. Then how this level of nitrogen will be maintained in atmosphere? For that question, we have answer in the topic called nitrogen cycle. What is nitrogen cycle? What is cycle? If it has a certain start point and it will revolve around and come back to same point as the wheel of the cycle you have seen, which point goes up, it will come down, it will go again. It is a continuous process and it is a cyclic process. Nitrogen cycle. In this, what will happen? Atmospheric nitrogen will be fixed by two means. One is by lightning. Lightning, as it gives high energy, nitrogen will be fixed. In that case, only 10% of nitrogen fixation is done by lightning. Majority is done by nitrogen fixing bacteria. N2 fixing bacteria, that is like rhizobium, azoospermum, many are there nitrogen fixing bacteria. How they will fix? They will fix and make them a nitrogenous compound. Lightning will convert it to nitric oxide, or they will be converted to nitrite, nitrate, ammonia. These are the nitrogenous compounds. These compounds will be used by plants. Plants will uptake these nutrients for their growth. They will give out cereals, crops. Nitrogen is very important in plants and animals to produce their nucleic acids, to produce their amino acids and proteins, building proteins, and producing carbohydrates also need nitrogen. Nitrogen is in every biomolecule, almost in every biomolecule. So it is very important. So plants take up nitrogenous compound and produce their proteins, etc. and it will be consumed by animals. Animals, right? Animals will consume plants and get those nitrogen rich compounds like proteins and all. And when the animals put their excreta out, or if they are dead, if the animals leave excreta or if they are dead, that will be again gone into soil, right? It will again reach the soil. In soil, it will be decomposed to form nitrogen compound. The complex biomolecules will be decomposed to NO2, NO3, that is nitrite, nitrate and ammonia. And this nitrogenous compound will not remain as such in soil, as we have to maintain amount of nitrogen in atmosphere. There are bacteria called denitrifying bacteria which will help the process of denitrification. Denitrification means converting nitrogenous compounds into gaseous nitrogen. Denitrifying bacteria will convert the compounds of nitrogen into free nitrogen, gaseous nitrogen in this process. In this process, the atmospheric nitrogen level will be maintained. As we know, there is almost 75 to 78% of nitrogen in our atmosphere. If that should be maintained, this denitrifying bacteria, the denitrification process should be possible, or it should be going on. So this was the end of the topic. There is also use of biofertilizers like rhizobium rich biofertilizers, and other are zoospirillum. Many microbes are used as biofertilizers to enrich the soil fertility by fixing nitrogen, gaseous nitrogen in the soil. So this was the topic for our today's session, and for this chapter microorganisms, friend and foe. Hope this chapter and this session have given some knowledge and helped in your studies. If you like this video, please like and share it to other friends, and please subscribe aksharaaalaya so that our upcoming videos will reach you and educate you. Thank you for hearing. Thank you.